Let us start with pedigree analysis. In humans, controlled crosses are not possible, so the study of family history about inheritance is used. Such an analysis of traits of in several generations of family is called pedigree analysis. The representation or chart showing family history is called the family tree or pedigree. In human genetics, pedigree study is utilized to trace the inheritance of a specific trait, abnormality, or disease. Now we can see the symbols that are used in pedigree analysis. Further, we will discuss genetic disorders. Genetic disorders are of two types: chromosomal disorders and Mendelian disorders. In chromosomal disorders, we have Down syndrome, Klinefelter syndrome, Turner syndrome, and in Mendelian disorders, we have hemophilia, sickle cell anemia, phenylketonuria, color blindness, cystic fibrosis, and thalassemia. We will discuss some Mendelian disorders. Mendelian disorders. The disorders caused by alternation or mutation in the single gene is known as Mendelian disorders. The pattern of inheritance of Mendelian disorders can be traced in a family by pedigree analysis. Mendelian disorders may be dominant or recessive. By pedigree analysis, it is easy to understand whether the trait is dominant or recessive. The representative pedigree analysis of autosomal dominant trait, example myotonic dystrophy, and autosomal recessive trait, example sickle cell anemia. The four types of mode of diseases are autosomal disease, in that we have dominant and recessive, and X-linked diseases, in that we have dominant and recessive. Autosomal dominant disease, they do not skip generation. Affected parents can have unaffected children. Example, Huntington disease, achondroplasia, dwarfism. Recessive, they skip generations. Unaffected parents can have affected children. Example, cystic fibrosis, sickle cell anemia. Next, we move on to X-linked diseases, where dominant we have in dominant we see that the transfer is never from father to son. All daughters of affected father will be affected. Disease never transfers to son. Example, vitamin D resistant rickets. Recessive, we have male are more affected. Disease tends to transfer from mother to the son and father to daughter, from father to daughter. Example, hemophilia and color blindness. First, we will see hemophilia. Hemophilia is a sex-linked recessive disease. In this, a protein involved in the blood clotting is affected. A simple cut results in non-stop bleeding. It is controlled by a pair of allele, H and N. H is a normal allele. Small h is responsible for hemophilia. Factor 8 and factor 9 are affected. Here, the heterozygous female carrier for hemophilia may transmit the disease to sons. The possibility of a female becoming hemophilia may transmit the disease to sons. The possibility of a female becoming a hemophilia is very rare because mother has to be at least carrier and father should be hemophilic, unviable in the later stage of life. An example, Queen Victoria was a carrier of the disease, so her family pedigree shows a number of hemophilia descendants. Sickle cell anemia. This is an autosome-linked recessive trait. It can be transmitted from parents to offspring when both the parents are carrier of the genes or heterozygous. The disease is controlled by a pair of alleles, HBA and HB5. The homozygous dominant HBA gives us normal, while heterozygous HBA-HB5 will be a carrier and will show the trait. Homozygous recessive will be HB5 and will be affected. The disease is caused by substitution of glutamic acid by valine at the 6th position of globin chain of hemoglobin. This is due to single base substitution at the 6th codon of the beta globin gene from GAG to GUG. The mutant hemoglobin molecule undergoes polymerization under low oxygen tension, causing the change in shape of the RBC from biconcave disc to elongated sickle cell structure. Thalassemia. In thalassemia, it is an autosomic linked recessive blood disease. It is transmitted from parents to the offspring when both the parents are unaffected carrier for the gene or heterozygous. It is due to mutation or deletion. It results in reduced synthesis of one of the globin chains, beta and alpha chains that make up hemoglobin. This forms abnormal hemoglobin molecules resulting into anemia. Thalassemia differs from sickle cell anemia. Thalassemia is a quantitative problem, synthesis of few globin molecules. Sickle cell anemia is a qualitative problem, synthesis of an incorrectly functioning globin. The two types of thalassemia are alpha and beta. In alpha, we see that the production of alpha globin chain will be affected. It is controlled by two closely linked genes, HBA1 and HBA2, on chromosome 16 of each parent. It is due to mutation or deletion of one or more of four genes. The more genes affected, the less alpha globin molecules produced. In beta thalassemia, we see production of beta globin is affected. It is controlled by a single gene HBB on chromosome 2 of each parent. It occurs due to mutation of one or both of the genes. Next we have phenylketonuria on 12th chromosome, an inborn error of metabolism, autosomal recessive trait. It is caused by mutation in the gene that codes for the enzyme phenylalanine hydroxylase, single gene mutation. The affected individual lacks this enzyme that converts the amino acid phenylalanine into tyrosine. As a result, phenylalanine accumulates and converts into phenyl, pyruvic acid and other derivatives. They accumulate in brain, resulting in mental retardation. These are also excreted through urine because of poor absorption by kidney. Further, we will see chromosomal disorders. The disorders caused due to absence or excess or abnormal arrangement of one or more of chromosomes are chromosomal disorders. They are divided into aneuploidy and polyploidy. Aneuploidy is the gain or loss of chromosomes due to failure of segregation of chromatids during cell division. It includes nalisomy, which is 2n-2, where a chromosome pair is lost from a diploid set. Monosomy 2 and -1, a chromosome is lost from a diploid set. Trisomy 2 and +1, a chromosome is added to a diploid set. And tetrasomy 2 and +1, where two chromosomes are added to a diploid set. Polyploidy, on the other hand, it is an increase in a whole set of chromosomes due to failure of cytokinesis after telophase stage of cell division. It is often seen in plants and very rare in humans. Now we'll see Down syndrome, Mongolism. It is the presence of an additional copy of chromosome number 21, that is the trisomy of 21. Genetic constitution is 45A plus XX or 45A plus XY, that is 47 chromosomes. The features are short stature with small round head, furrowed tongue and partially open mouth. Palm is broad with characteristic palm Cs. Retarded physical, psychomotor and mental development. Congenital heart disease. Next we'll see Klinefelter syndrome. It is the presence of an additional copy of X chromosome in male. Genetic constitution 44A plus XXY, that is 47 chromosomes. The features of Klinefelter syndrome is overall masculine development, however feminine development of breast, that is gynecomastia, is also expressed. They will be sterile and mentally retarded. Turner's syndrome, we see that it is the absence of one of the X chromosome in female. The genetic constitution here is 44A plus XO, that is 45 chromosome. The features of this are sterile ovaries are rudimental, lack of other secondary sexual characters, they are dwarf and they are mentally retarded.